For this discussion, I'm going to do a walkthrough of the Charlesworth and Charlesworth paper. I think it's worthwhile. Hopefully you've read it. If not, this will be a good review. It does a really good job at establishing some fundamental kind of foundational principles of population genetics and giving you a history of them. We'll get a little bit more into depth in coming lectures, but not a lot. Population genetics is a very statistically intense and mathematically intense subject. We could take an entire semester. Well, I couldn't. I'm not an expert in the pop gen area, but an instructor could and could take an entire semester and get really in depth. We're just going to do a brief section and give you an overview of this, but there are some important highlights here that some of which you may have come across in previous classes and others that will be new to you. So I'm going to make some notes on this. It'll be a little bit kind of like a chalk talk, barring the ability for you to jump back in and participate. But the first thing that I want to point out as we're talking about this, the vast majority of this, particularly in the early decades, was all done in Drosophila. Okay, so we're talking about fruit flies and that they are really, can't write really well here, but let's see what we can do. Drosophila, right? And melanogaster, but then expanding out into other species. And today, Drosophila still is the major workhorse for population genetics, other areas of molecular biology also. Okay, so just be aware of that. The first thing that I want to point out is this early division, disagreement between the people that were beginning to work. Now, really, this is about 100 years or so after Darwin's theory of natural selection comes out. And so really there was a lot of talk and some progress, but not a lot was made until we begin to have the molecular tools to look at some of this diversity and variety. And so we're just seeing the beginnings of that really in the 1960s. And so we have this dichotomy, if you will, between what is called the classical view, underline it there, and the balance school of variability. Now, basically, these two ideas, and again, this was before we had lots of data, the classical view was that for most loci, most genes, that we would see one major beneficial allele, and then maybe a few other alleles that were there in very small frequencies, but they were negative and were slowly being removed by natural selection. So this classical view kind of held more to the old Darwinian view that natural selection has a clear winner for each gene, and then it works to remove, quote unquote, the losers. Now, the balance school said that there were other more complex forces at play, and so we should see more genetic diversity with higher frequencies for some of the other alleles. So there might be two beneficial ones. There might be advantage to maintaining diversity at an allele, and so natural selection works to keep diversity rather than to remove everything except for one variant. And so this was a very early kind of disagreement. And by and large, this balance school seems to be more, although there are certainly genes where the classical view takes on. And then shortly after that, people began to start thinking about what we would term the neutral. And one of the amazing things about this is that it really occurred before we had much data to back it up. Pardon my handwriting. I'm trying to write with a mouse, and this is not the ideal way to do it, but maybe it's legible at least, right? So this neutral idea was that most of the diversity at the DNA level is going to be effectively neutral. It's not going to be acted on either positively or negatively by natural selection. And again, this was proposed before we had large data sets. And then we began to get DNA data, well, first molecular data, not really DNA based, but molecular tools that allowed us to evaluate this. And I'm going to take a minute, maybe then we'll jump back a little bit, but I want to talk about kind of a progression of the molecular tools that were available. And this is really on pages 3 and 4 of this Charleston paper. So I'm going to skip down to 4, and we will make a list here of the molecular tools that were available, kind of right over here to the side. Okay, so the first molecular tool that was really available for us to evaluate things was gel electrophoresis of alleles. Now, hopefully you are already familiar with the process. If not, I'll outline it briefly. You do need to be familiar with this and understand how this was used. So gel electrophoresis is when you put molecules, and we use it for DNA and RNA also, but make sure I get it spelled here, these long Latin and Greek base words, okay? Gel electrophoresis. And basically what you do is you put a sample of your isolated molecules on a gel, and then you run it. You can run it in one direction or even in two directions. One may be based on electrical charge and the other based on some other gradient like a salt gradient or a pH gradient or something like this. And that way you can separate these molecules based on their different physical properties. And so you could isolate different alleles. So if you were able to get all of the alleles for one gene and then run them on a gel, you could isolate them out on this electrophoresis. They would separate out differently, and you could tell how many different alleles there were, and also how much of abundance they were in your sample based on how bright they were on the gel. Okay, so again, I didn't get into a ton of the details, but hopefully you're somewhat familiar with gel electrophoresis. Now, there are two main drawbacks that the paper points out to gel electrophoresis. So let's make that, you should be aware of these. So let's put drawbacks. Okay, the first one is number one, and I think they do these in different orders, but I'm gonna put the first one here, is that this is at the amino acid level and not the DNA level. So basically what this means is we are going to be unable to detect any DNA changes that don't change the amino acid. And we realize that that actually incorporates a large amount of the diversity for these alleles. And so although it does capture much or maybe even all of the functional difference between genes, it doesn't capture the difference at the DNA level. So just as an example, you should already be familiar with this from previous lectures, but a CCC codon codes for proline, and if we get a mutation, that is a CCA. So now we have polymorphisms in this population. Right? Some individuals carry a CCC variant, some carry a CCA. Gel electrophoresis will not be able to capture that because both of these are a proline in the resultant amino acid sequence in the protein sequence. Okay, so that's drawback number one. Drawback number two is that even amino acid differences, so we'll put amino acid differences, and I'm going to abbreviate here a little bit because of my difficulty in writing here. It looks a little bit like a T. Even amino acid differences may not change the way they migrate on a gel. Let's put may not change migration. Okay? So sometimes, like an isoleucine for a leucine, which are two very chemically similar amino acids, a change, or maybe even multiple changes, they may still, for whatever reason, clump together. So although gel electrophoresis was our first step forward, it was very limited in what we could do. Okay? So the next major step forward came with the ability to do things at the DNA level. And the first one that was present for many years and was where much of our original data came from was using restriction sites. Now, a restriction site is an area of DNA that is recognized by a restriction enzyme. And basically, restriction enzymes are proteins that cut DNA at very, very specific points. So what we could do is we could take two DNA sequences. I'll draw lines here to represent the DNA sequences. And if there were mutations in there, even though at this point we couldn't sequence the DNA, right? These mutations, which made one sequence different from the other, if it was in an area that was recognized by a restriction site, then when we added restriction sites to these two different sequences of DNA, so this might be variant number 1, this might be variant number 2 in the population, and even though they might be identical phenotypically, they may even have the same amino acid sequence, they may be cut differently by a restriction site because of these quote-unquote hidden or silent mutations that aren't picked up by the protein level. And so when this was done, we had another new, fairly rich source of DNA. And again, much of this work was done in Drosophila, but we could take all of them and cut them up and say, oh, look, some of them, when we chew them up with one of these restriction enzymes, we get pieces that are, of length like this. And maybe the other one, we're missing a restriction site there, and so we get only 2 pieces, right, that look like that because we're missing one of the restriction sites. So that's a way to pick up variation that we couldn't see before. Okay, so progress, right? We're now able to look at the DNA. So this one can pick up not all, because our restriction profile is limited. There are many different restriction enzymes, so we could do many of them, but it's very labor intensive. And even with all of them, we might be missing some of the restriction sites. Or some of the differences may not result in restriction sites. So we're beginning to capture information at the DNA level, but still not able to capture all of it, right? So the next step forward was PCR, and there were a few methods briefly available before PCR that were used, but the next method we're going to say PCR and DNA sequencing. And more particularly, it's what we're going to call Sanger sequencing. And this was used for a couple of decades, was really a huge major advance, but was very labor intensive and was only targeted. We had to target a single specific gene, do a lot of work, and then we could get variants for that gene. But this allowed us to now actually look at the actual DNA sequence. So we could get the entire sequence, A, T, G, C, and so on down the line, right? That entire DNA sequence for one individual. We could sequence it for another individual, see where it was different, right? So maybe somewhere down the line we have a T at one position and A at the other, and we can recognize that as a mutation, whether or not it leads to a difference at the amino acid level. So this was a huge breakthrough. Now we're able to look at down the DNA sequence. We can't look at the entire genome. And so what people did was they targeted genes of interest and areas of interest and worked really hard on those, but we were unable to look at the entire genome. So our final step and breakthrough, which is kind of what we're in the middle of right now, was genome sequencing, Bailey. Sometimes people say next generation sequencing, but we'll just say genome sequencing. So when we are now able to, basically using different methods, they're related to these early methodologies, but different methods to do entire genomes. And so now not only do we have these short little pieces of DNA that we can compare, right? So we could say, oh, maybe earlier we'd look at gene 1, and then maybe we'd do a lot more work and a lot more energy and look at gene 2, and then we'd be able to say something that's kind of cool about, what's going on with these specific examples. But today, with genome sequencing, we can sequence entire genomes over a wide variety of individuals in a single population. We can then look at individuals in another population. So we're generating these massive data sets where not only are we doing gene 1, 2, 3, maybe a handful of genes, we have thousands of genes and thousands of samples in those genes. So we can really begin to look at the genetic diversity of species. And that's really where we're at. So that's kind of the progress of the methodologies. Okay, so be familiar with those 4 steps and kind of what each one was capable and able to do. And each of them was an important step forward, right? But now we have this incredible amount of information. And one of the things that came out of this that was really useful, especially as we're getting these large data sets, was our ability to compare these silent mutations, right, that do not affect phenotype, and the ones that are subject to natural selection, right? That are not silent, they're either good or bad, and so natural selection can work on them, and we can begin to compare them. Now, I want to make a distinction between 2 types of silent mutations. I hope you're familiar with these. When I say silent, it simply means it does not impact the phenotype of the organism. And we're going to look at 2 major categories. The first one is within the protein coding sequence. And these most commonly occur at the 3rd position in a codon. So these are called synonymous, right? But they're a type of a silent mutation because they result in the exact same amino acid, no difference at all in the phenotype of the organism. So 3rd position, synonymous. And I'll just abbreviate it here because I'm running out of space. So that's one important category of silent mutations. Another very, very important one is in non-coding DNA. Now, non-coding DNA can have a phenotypic impact. So not all mutations in the non-coding DNA are silent, but many of them are, because the phenotypic impact of the non-coding DNA is spread out over such a huge place. It's rather diffuse. So not many parts of that non-coding DNA are going to have a significant phenotypic impact if they are mutated. And so we open this entire new world of diversity and genetic variety that we can now see, that we can do entire genome sequencing projects. Okay, and so this led to this breakthrough of neutral theory, right? So I'm going to move on to the next page. That one's a little messy anyway, so it's good, right? Actually, there's one thing I guess I should put here that's pointed out in this paragraph here. And this we've already talked about, that there was a early hypothesis later supported by the data that the substitution rate should be proportional to the mutation rate because of the offsetting effects of population size. So mutation rate should be proportional, or we'll even say equal to, the substitution rate. And this is, of course, if we can infer neutrality. And for a vast majority of the genome, maybe third positions in general, and much of the non-coding DNA, we can infer neutrality because there's no force of natural selection acting on these silent mutations. Okay, so I'm going to point out just a couple other highlights here as we move forward. So now we're getting a little bit more into the theory. A major advance came when we talked about coalescence. And coalescence is the idea that we can look at all of the diversity in a modern population, right? So maybe we sequence 18 individuals and we find a certain number that have the same variant represented by X, and others have a different one. We do this through DNA sequencing. And we can look at all of the variation present. This is just several, but maybe we do thousands. And then we can figure out that if they are evolving neutrally, how long would it take, how many generations back would we need to go before we had a single ancestral allele? And that's going to depend on the population size. Coalescence is faster in smaller populations. We'll look at that in our next lecture. And it's also going to depend on the mutation rate, right? How often do we see? So within a single population, now that we're not comparing between populations, population size is still critically important, and the mutation rate. And coalescence was, in essence, looking backwards in time. That was a major breakthrough. Okay. The next major point that they point out is this idea of a linkage disequilibrium, which is abbreviated LD. Now, linkage disequilibrium is the non-statistical association of two different alleles, that they're not completely randomly associated, which classical Mendelian genetics would assume that they're going to be completely mixed up at each generation. So for instance, if you have blue eyes, and blue eyes is controlled by one gene, there should not be any association between, say, blue eyes and blood type. And that's true unless those two genes are found right next to each other. So maybe the I gene, right, which we have a blue eye allele, and we'll just call it the Z gene. If they are right next to each other on the chromosome, they are going to be linked together. Now, this is an idea that should not be unfamiliar to you. This is done in introductory biology. But what that means is that most commonly, if these two alleles are found next to each other, they're going to be inherited together. It's only when we get a recombination event in the middle of them, right, that we get those two alleles broken up. So imagine we have everybody in the population we'll just, for our example, we'll start with this. Everyone in the population they're homozygous for the X allele, which the X allele for the I gene, which makes your eyes blue. So everybody maybe we're looking at a rabbit population. Every single rabbit is homozygous, and they all have blue eyes. And every single rabbit has the exact same blood type. And we'll do it, we did a Z, didn't we? So every single rabbit has this Z blood type. Okay, so no big deal. There's not any diversity and variety. This is unusual, but let's say we found it. And those genes were right next to each other. Now suddenly there's a brand new mutation. And so a rabbit gets a mutation that maybe makes it have brown eyes, let's say. And so we'll represent this mutation. So suddenly maybe originally they're heterozygous. Genetics might play a role here. But we have some rabbits that have a brown-eyed allele instead of this blue-eyed allele. But they still have the same blood type. Okay? So now suddenly we have a different genetic linkage. And maybe this brown allele is not really great, good or bad, and so it kind of spreads, and we get some that have blue eyes and some that have brown eyes, and they all have the same blood type, okay? And no big deal because the brown versus blue eyes didn't make a difference, and it's just maintained in some level in the population. But then suddenly we get a second mutation, right? So there's our first mutation right there. Later on in another individual, we have, and maybe they were heterozygous for this I gene, and we get a new mutation that gives them a new blood type. And so we'll call this one Y for the new blood type. And just by chance, that new blood type happens on a chromosome that carries the blue eye allele. So there is our second mutation. Now, let's say this one has a huge natural selection advantage, right? That is a good thing. Whereas before it was just kind of neutral, right? The eye color change didn't make a difference to these rabbits. But now suddenly we have a huge advantage. This blood type allows them to survive longer, have more babies, whatever. It's a huge advantage. And it just by chance, there wasn't any rhyme or reason to why it occurred. It was just a random mutation. It occurs by the blue-eyed allele. And so if this is selected for, it's a huge advantage and becomes very, very common. It is going to drag along this blue-eyed allele. And we're going to see the brown-eyed allele really, really go down in frequency from what it was before. Now, this is often called genetic hitchhiking. The technical term is linkage disequilibrium. Put my E in there. So genetic hitchhiking. So you should know what that is. And what this can result in is if we have a new mutation that's really, really advantageous, finish my hitchhiking here, is that this genetic hitchhiking can result in something called a selective sweep. And a selective sweep occurs when we have a really advantageous allele, either due to a brand new mutation or maybe a change in the environment that makes one of these alleles very useful. And so alleles that are next to it, so there's our advantageous allele on the genome, all of the alleles that are next to it, and the farther away, the less likely this is to happen because genetic recombination can break it up. But all of the alleles that just by chance happen to be next to it get dragged along, and so the end result is an entire population with very low genetic diversity in that region of the genome. Okay, low diversity. My handwriting really is better than this when I have a pen. So selective sweeps result from natural selection for just one allele, but all the other alleles coming along and making an area of the genome that has very, very low diversity. And so we can look at the genome as a whole, right, now that we have this DNA sequencing ability, and now we can begin to say, what is the role of recombination? And if we have areas that have very, very high recombination, they're not prone to these selective sweeps as much, right? So we can look at these recombination hotspots, which have very, very low levels of selective sweeps, and so therefore they would have higher diversity because they're recombination hotspots. And we can also maybe see how often a new mutation occurs that really kind of pulls other things along with it. Okay, so that's a selective sweep. It's kind of defined on this next page here, but make sure you're familiar with that idea and what a selective sweep is. Okay, now the next major breakthrough came with comparing within population and between population. And both the HKA and the McDonald-Kreitman test, which are outlined here, are just different ways to do a similar thing. And basically, this came by comparing the amount of diversity, originally just overall diversity, that's what this test does, and then making a distinction between the silent amounts of diversity, particularly in genes, so synonymous mutations versus non-synonymous mutations. But basically, here's how it works. If everything is evolving neutrally, and we are comparing two different species, so I'm going to do a little phylogeny here, species A, B, and C, and let's say that we want to compare the evolution that's occurred between species B and C. And I have a gene, and I don't know if that gene is evolving neutrally or if there's been selection in one or more of these lineages. And so what I could do with these tests is take many, many samples. So within species B, so these are all individuals within species B, and because it's within a species, we're going to have more of a network-like pattern, a complex set of relationships within that species. And I could look at all of the diversity that I find, do an adequate sample, and look at the diversity. We'll just call it variation within species B. Okay, so if I looked at the variation within species B, and then I did the same thing, got adequate samples, and again, much of this work was done in different Drosophila species. I looked at the variation within species C, right? What I would expect to see is that they would maybe be roughly similar if everything was neutral. Now that's the first step, but basically I would put these two together and say, what is the average variation? So we would look at intra-species variation, right? What is the average variation within these species? And if everything is neutral, then I would expect this intra-variation, right, intra-species variation to be roughly equivalent. So we'll draw a roughly equivalent equal sign to the interspecies variation. So unfortunately, these are very similar. Be careful. Intra within the species, inter between the species. So now I'm going to compare overall the difference between species B and species C, right? So that would be the variation here between them. So we'll call a variation, I'll just call it VI. Okay, whatever. That's not a common term, but the variation between them. If those are roughly the same, if they're equivalent, then that means that this is evolving neutrally. And so neutral theory in this application and in many others, neutral theory becomes our null hypothesis. Right? So that is kind of a way to begin to start. And then if we see variation, if these are not statistically similar, if they're not the same, and there's a statistical test, you do a chi-squared test and test to see whether or not they're significantly different than one another. That's beyond what we're doing in this class. But if they are significantly different, then suddenly we have, oh, there must be selection, and we begin to do more work. Is the selection in that lineage? Is the selection within this species? Or maybe the selection happened as that species diverged. Okay, and so we can begin to look at this. And then the last point that I want to point out. So just remember, neutral theory is kind of our null hypothesis, and when we see violation of neutral theory, we'll look later at other ways we can test that. But just no McDonald-Kreitman test and this HKA test are ways to do within species versus between species variation. The last point that I want to highlight here is that there has been major breakthrough with behavioral biology. And they point this out, especially with regards to kin selection. But behavioral biology, we can now quantitatively do much of the behavioral biology. Right? So we can look at both kin selection, and the other main advantage was game theory and this idea of an evolutionarily stable strategy. Okay, you should be familiar with those from previous classes. If not, just look up a definition. That's fine. I won't go into it here. But just be aware that those are two applications of our now quantitative assessments of diversity and looking how they affect behavioral interactions. Okay, I misspelled behavioral. That looks weird to me, right? That should be an I. Oh, that's going to bother me. There, I fixed it. But behavioral biology. Okay? All right, so that's just basically a walkthrough, talk-through. In our next discussion, I'm fusing units 2.8 and 2.9 onto one day because I've had some research that has interrupted my schedule here. But I posted a new calendar, so take a look at that. I'll have everything up by the end of the day on Saturday for Monday's material. And of course, on Wednesday of next week, we have our second exam.